This video will be detailing the citric acid cycle, which is also known as the tricarboxylic acid, or TCA, cycle, or the Krebs cycle, named after the scientist who discovered it, Hans Krebs. To begin, we show a broad overview of cellular respiration and its three distinct stages. In stage 1, which occurs in the cytosol, some fatty acids and some amino acids are oxidized to acetyl-CoA, and glucose is converted to pyruvate via glycolysis, which also gets oxidized to acetyl-CoA. Acetyl-CoA then gets transported into the mitochondria, beginning stage 2 of cellular respiration, the TCA cycle. During this stage, acetyl-CoA is oxidized through a series of cyclic reactions to shuttle electrons to the third stage, the electron transport chain, or the ETC. In stage 3, the electrons are shuttled through the mitochondrial membrane by a chain of electron carriers to ultimately produce ATP. Also shown here is a legend explaining the color coding that will be seen throughout this video. This video will be focusing only on stage 2, the citric acid cycle. This cycle is considered so vital to cellular functions that mutations or deficiencies in it were long considered to be fatal in very early development. The citric acid cycle starts with the condensation of acetyl-CoA and oxaloacetate to produce citrate, and then through a series of several other reactions, regenerates oxaloacetate to restart the cycle. Let's quickly review glycolysis, where glucose is converted through a series of reactions to produce 2 pyruvates, 2 NADH, and a couple of ATP. The resulting pyruvate is then converted to acetyl-CoA through oxidative decarboxylation, catalyzed by a multi-enzyme complex called pyruvate dehydrogenase complex, or PDH. A mutation in this enzyme can have severe consequences, resulting in an inability to oxidize pyruvate normally. This can be particularly catastrophic for cells whose main source of energy comes from the oxidation of glucose, such as brain cells and red blood cells. The PDH complex consists of 3 enzymes and 5 cofactors, and transforms pyruvate to produce acetyl-CoA, CO2, and NADH. While technically not considered a part of the TCA cycle, the oxidation of pyruvate to acetyl-CoA is an essential step and one of the main pathways for the production of acetyl-CoA.
Now let's begin discussing the individual reactions of the citric acid cycle, beginning with the formation of citrate. The enzyme citrate synthase catalyzes the condensation of acetyl-CoA and oxaloacetate to produce citrate. In this reaction, the 2-carbon acetyl group of acetyl-CoA is transferred to the carbonyl group of the 4-carbon oxaloacetate to produce a 6-carbon citrate. The intermediate citral-CoA is formed and is very quickly hydrolyzed to produce free CoA and citrate. This quick hydrolysis is highly exergonic and is what drives the reaction forward, making it a regulatory step. Next is the transformation of citrate to isocitrate, both a 6-carbon molecule.
This takes place in a 2-step reaction with the enzyme aconitase catalyzing the reversible dehydration of citrate to the intermediate cis-aconitate and forming a double bond. Aconitase then catalyzes a second reaction, this time a hydration of the double bond, to form isocitrate. Isocitrate is rapidly consumed in the next step, driving the reaction forward. Next is the transformation of isocitrate to alpha-ketoglutarate, which also forms the products carbon dioxide and NADH. The enzyme isocitrate dehydrogenase, which has 3 forms, catalyzes the oxidative decarboxylation of isocitrate, a 6-carbon molecule, to form alpha-ketoglutarate, a 5-carbon molecule. Isocitrate binds to the enzyme and is oxidized by hydride transfer of the coenzyme NAD+ to NADH or NADP+ to NADPH, depending on the form of the enzyme. A manganese ion then binds to the carbonyl group of the intermediate oxalosuccinate, allowing for the loss of a carboxyl group as CO2 and then the rearrangement of some electrons yields the alpha-ketoglutarate. In the last regulatory step of the citric acid cycle, alpha-ketoglutarate is converted to succinyl-CoA. This step is another oxidative decarboxylation, in which CoA, NAD+, and a multi-enzyme complex called alpha-ketoglutarate dehydrogenase converts the 5-carbon alpha-ketoglutarate to the now 4-carbon succinyl-CoA and CO2. NAD+ serves as the electron acceptor and CoA as a carrier of that succinyl group that forms the thioester bond of succinyl-CoA. The alpha-ketoglutarate dehydrogenase complex is very similar to the PDH complex, shown here. They both consist of 3 enzymes, E1, E2, and E3, and 5 cofactors. The enzymes in both complexes are homologous to each other, and the cofactors, FAD, NAD, TPP, lipoate, and coenzyme A are the same in both. E1 of both complexes are structurally similar, except for changes due to the binding specificity of the substrate. Alpha-ketoglutarate for the alpha-ketoglutarate dehydrogenase complex, and pyruvate for the PDH. E2 and E3 in both complexes are very similar to each other, with little to no differences among the two. Next is the conversion of succinyl-CoA to succinate. In this energy-conserving reaction, succinyl-CoA synthetase catalyzes this reversible reaction with help from its cofactors GDP and inorganic phosphate. Succinyl-CoA binds with the incoming enzyme and an inorganic phosphate to form the enzyme-bound succinyl phosphate intermediate. Succinate then comes off and the enzyme gets phosphorylated at its histidine residue. A GDP molecule then comes in and the enzyme transfers the phosphate to GDP, forming GTP. The use of this inorganic phosphate conserves energy by not sacrificing an ATP for its phosphate group.
The remaining reactions, all 4 carbon molecules, serve to oxidize succinate back to oxaloacetate for another round in the cycle. The following reaction is the formation of fumarate from succinate. Succinate dehydrogenase catalyzes the oxidation of the central single bond in succinate to a trans double bond forming fumarate. FAD in this case serves as the electron acceptor, getting reduced to FADH2. Next is the formation of malate from fumarate. The formation of malate is a hydration reaction catalyzed by fumarase to yield L-malate. Fumarase is highly stereospecific. It hydrates the trans double bond of fumarate, but not the double bond of maleate, which is a cis isomer of fumarate. The last step in the TCA cycle is the reformation of oxaloacetate from malate. This step is catalyzed by the NAD-linked L-malate dehydrogenase. Under standard conditions, this reversible reaction typically lies to the left, but the rapid consumption of oxaloacetate keeps the concentration low, driving the reaction forward.
Now that we have made a complete turn of the cycle, let's take a look at what regulates it. There are 3 regulation points in the citric acid cycle. 4 if you begin with the transformation of pyruvate to acetyl-CoA prior to its entrance into the TCA cycle, as we show here. The production of acetyl-CoA from pyruvate by PDH is regulated allosterically and by covalent mechanisms. The reaction is activated when energy demand is high and there are increased levels of AMP, CoA, NAD+, and calcium. It is strongly inhibited by ATP and by acetyl-CoA and NADH, the products of the reaction. The conversion of pyruvate to acetyl-CoA is also inhibited by an abundance of fatty acids present. The following true regulatory steps occur in 3 highly exergonic reactions of this cycle. They are those steps catalyzed by citrate synthase, isocitrate dehydrogenase, and alpha-ketoglutarate dehydrogenase. Citrate synthase is inhibited by increased NADH, succinyl-CoA, and ATP, while high concentrations of ADP will activate the reaction. Isocitrate dehydrogenase is inhibited mainly by ATP and activated by calcium and ADP. And finally, alpha-ketoglutarate dehydrogenase is activated by calcium and inhibited by its products, succinyl-CoA and NADH. The availability of substrate can also affect the progression of the cycle. For example, low levels of acetyl-CoA and oxaloacetate can limit the rate of citrate formation. The primary role of the TCA cycle is the production of electron carriers that drive the downstream production of ATP in the third and final stage of cellular respiration, the electron transport chain. This production of ATP helps to maintain homeostasis by supplying the much-needed chemical energy a cell requires to function properly. In addition, the TCA cycle also serves as an important supplier of biosynthetic precursors critical to those cellular functions that help maintain homeostasis, such as amino acids, fatty acids, and heme. This image serves to highlight some important anaplerotic reactions, shown with red arrows, that can convert products back into intermediates in the TCA cycle. This image also showcases a broad overview of how important the TCA cycle is to overall cellular function, and that without it, aerobic life could not exist.